All right, everybody, this is chapter 10 in Krog, and in this chapter we're going to be learning about meiosis. So in the previous lecture we learned about mitosis, and mitosis is done for cellular growth or for organismal growth. It's done for repairing things. So if you get a cut in your skin, the process of replenishing those damaged cells is going to be mitosis. So this is meiosis, and they're very similar sounding things, but they are very different processes. Meiosis only makes gametes. That's an important thing to remember. Meiosis only makes gametes. It only makes, in human beings, sperm and eggs. Mitosis is the copying of lots of different cell types. Meiosis only makes sperm and eggs, only makes gametes. So, again, like I was just saying, what is mitosis for? What is the purpose of mitosis? Well, it's for growth and repair. If you want to get bigger as an organism, you're going to need to use mitosis. If you need to repair something, again, you use mitosis. Meiosis, what is meiosis for? Meiosis is to create those gametes. And as we're going to see in a moment, the whole point of those gametes is to increase genetic diversity so that we can reproduce sexually. And sexual reproduction allows these combinations of traits to come together, and it also increases the diversity of a group of organisms, which is going to be advantageous. In biological systems, diversity is always advantageous. Well, I shouldn't say always, because nothing is always true in biology, but diversity is going to help things to be able to survive. If, for instance, they have some shift in their environment, increased diversity is going to help those organisms to be able to stick it out and last for longer. And one of the ways that organisms increase their diversity is through this process of sexual reproduction. So why meiosis? We want to increase our genetic diversity without increasing the number of chromosomes in our offspring. So N, we're going to see this later on, N is equal to the number of chromosomes in an organism. So you're a human being, and you should know that you have 46 chromosomes. So you are diploid, you are 2N. So your diploid or your ploidy number would be 2N equals 46. That might sound like kind of a lot, but the basics of it here is N is just the number of chromosomes, and because you have two copies, you have two sets of chromosomes. You got one set of chromosomes from your mom, you got one set of chromosomes from your dad. You are 2N, and so again, your chromosome number is 2N equals 46. If you took a somatic cell from a woman, and somatic just means body cell, so we're not talking about sperm and eggs right now. We're just any body cell not other than sperm or eggs, but any body cell, so not a germline cell. If you took a bit of skin from a woman and you took a bit of skin from a man and you stuck them together, you would have 92 chromosomes. This individual would be 4N. Okay. If you repeated this process, and that 4N person had kids, there would be 8N equals 184. And that number would just continuously increase forever. And we would just wind up with people that had thousands and thousands of chromosomes, and I'm sure you can see why that would not work. So what you have to do is you have to decrease the number of chromosomes before you reproduce. And the way that we do that is using meiosis. Okay? We reduce the number of chromosomes using this process of meiosis. So cells with half the chromosome numbers are called haploid, and these are called, in humans, gametes. So this is an important term to know. Haploid is half the number of chromosomes normally needed by an individual, and those are sperm and eggs. Okay? Those cells are made only by sexually reproducing organisms, and only in a special organ called the gonads. We're going to learn later on about different organisms that use different strategies other than sexual reproduction to reproduce. We already learned about one. We learned about the binary fission of bacteria. And the process by which a single diploid cell divides to produce haploid reproductive cells is meiosis, like I've been saying. So let's think about this really quick. We have 46 chromosomes. The person we want to reproduce with has 46 chromosomes. But at the end of this reproduction, we want our offspring to also have 46 chromosomes. So you can't just throw cells together. You can't just throw genetic information together. You need to be able to reduce that genetic information by half and get to these haploid cells. So meiosis reduces the chromosome number by 2 divisions instead of 1. We're going to see how this process plays out. So if you're not just like memorizing things, that now it's not that big of a deal. So in mitosis, and you're going to see a lot of comparing and contrasting of mitosis and meiosis. In mitosis, identical DNA is duplicated. You wind up with 2 sister cells that are identical to one another. They have the same ploidy number. They're both diploid, 2N. Meiosis is reductional, so you are winding up with half of the chromosomes than you actually started out with. There are 2 separate cell divisions in meiosis, whereas in mitosis there is only 1. So meiosis is separated into meiosis 1 and meiosis 2. And so 1 haploid cell becomes 4, or 1 diploid cell becomes 4 haploid cells. We're going to talk about that process. So again, this is setting you up and comparing and contrasting mitosis and meiosis. So in mitosis, as we learned, your chromosomes duplicate. So now you have these copies of these chromosomes, and then the chromosomes actually wind up getting separated out. And so after this first division, you wind up with cells that are all identical to one another. All right, and this image here is talking about these homologous pairs. Let's say this blue copy is from your mom, okay? This is her set of chromosomes that she gave to you. And this red set over here, this is from your dad. This is the set of chromosomes that your dad came from you. These are homologous, which means they have the same size, they have the same functions, they code for similar proteins. These are not identical, but they are similar to one another. Again, this is the one from your dad, this is the one from your mom. They have similar genetic patterns on them. And in mitosis, you wind up with exactly what you started with. In meiosis, you wind up with 1N cells, or they're also known as haploid cells. Right? They're duplicating their DNA, and then they're having 2 divisions. And we're going to go through this process a bit here. So meiosis begins just the same way that mitosis does, with replication of DNA in the S phase of interphase. There is interphase for these meiotic cells as well as mitotic cells. Okay, so we start with meiosis 1. We have these homologous pairs that have been duplicated. I'm going to show you a picture if you can't really picture this quite well. But you have these homologous chromosomes, each of which has duplicated into a sister chromatid. Okay, you have homologous pairs, each of those duplicate, each of these now has a sister chromatid. That sister chromatid is going to come together into one piece of DNA, and this magical thing called crossing over, which is the process of recombination, is going to happen. And again, if this is crazy confusing, it's okay, we're gonna talk about it a bit. The next phase after prophase 1 is metaphase 1. Those tetrads I was talking about earlier are going to line up on the metaphase plate, and they're also going to independently assort, which I'm going to talk about later. Then you have anaphase 1, which is where the chromosomes, or the homologous pairs rather, are going to separate to opposite poles. And then telophase 1, which is where those cells are going to pinch off from one another and create 2 new haploid cells. So here you have the end of interphase. DNA has replicated in there. So if you could picture the homologous pairs, right? Each of those, there's now 2 copies of each of those homologous pairs. Then you have prophase 1, where those 2 sets of homologous pairs are going to link up to one another and do what this thing called crossing over. And in crossing over, they're going to be exchanging little bits of genetic information with one another. As you can see here, this chromosome's got a little piece of red on it, and this chromosome has a little piece of blue on it. That's the process of crossing over. They're going to line up here on the metaphase plate, and then during anaphase, they're actually going to separate from one another. So again, here's some more details about crossing over. You can imagine before these duplicated, you just had one red one, and then you had one blue one, right? Then during S phase of interphase, these chromosomes duplicated, and you wind up with what is a sister chromatid. A sister chromatid. These two pieces of DNA are identical to one another. These two pieces of DNA are identical to one another as well. So the blue one, these two blue sets are identical. These two reds are identical. And this red one is not identical to this blue one. They have different genes, like I talked about earlier. You got this blue one from your mom, you got this red one from your dad, or whatever it is I said, whichever order. Okay, they're not identical to one another, but they're similar. They have some of the similar genes. Then during prophase 1, they get together and form this tetrad, and they undergo crossing over, wherein some of the genes from the blue one attach to some of the genes from the red one, and some of the genes from the red one jump onto some of the genes from the blue one. It's incredible. This is two people's, or this is your genetic information switching places with one another. It's absolutely fantastic. So after metaphase 1, or during metaphase 1, you have this process of independent assortment. I'm planning on drawing this on the board just because it can be a little bit difficult to think about in a lecture in this format. But you can see here, these chromosomes could have lined up like this with a red one here and a blue one here. Or they could have lined up like this with a red one here and a red one here. There's nothing preventing either of these chromosomes from being in either order. And so when they line up in these different ways, you wind up with different gametes at the end. You wind up with different combinations of red and blue. That process is called independent assortment. Independent assortment is happening here in metaphase 1. And like I said, I'm going to do this again on the board just because it's a kind of an important idea and it's a little bit difficult to think about. Just want to make sure everyone gets that. So the first way that we've gotten increase in genetic information is not genetic information. The first thing we've gotten an increase in genetic diversity was via crossing over, and now independent assortment is adding more diversity to this. So after these processes have happened, which you'll notice looked similar to mitosis but with some differences, you have sister chromatids, those exact copies of one another, which have undergone crossing over, which means they're not so exact anymore. When you enter prophase 2, which 23 identifiable separate chromosomes, still with 2 sister chromatids. Prophase 2 is kind of a boring stage, not a ton happens. I'm sure some molecular biologist is probably screaming right now, or they would be if they could hear me, about all the interesting stuff happening in prophase 2. I just don't know what interesting stuff is happening there. But metaphase 2, those chromosomes are lining up on the metaphase plate, this time with a sister chromatid on either side. And then during anaphase 2, those sister chromatids are going to separate. And in telophase 2, you're going to form 2 nuclei. Cytokinesis, or the splitting or the pinching off of the cell, is going to begin, and you're going to wind up with a total of 4 haploid cells at the end. So here's the telophase 1, telophase 1. This is the end, right? All that stuff we just talked about has happened. Here's prophase 2, brief, aka boring. Then you have metaphase 2, in which these sister chromatids are lining up along the metaphase plate. And then anaphase 2, where they're actually splitting. And then cytokinesis, you're winding up with 4 haploid cells that are unique. They're all different from one another because of crossing over and independent assortment. So every time this process happens, you're going to produce 4 gametes, okay, 4 sex cells, 4 cells that are ready for reproduction. So the problem of chromosome duplication is certainly solved by meiosis, because remember, if you took my 46 pairs and put it together with another 46 pairs, that'd be way too many sets of chromosomes. You also create diversity, which is the most important function of meiosis, okay? Because of recombination, where genes are exchanged with one another, and independent assortment means that you wind up with unique combinations at the very end. This has a ton of significance for genetic diversity. Like I was saying, this is a great way to drive genetic diversity in a system or in a population, because you are increasing the diversity of all those gametes from those original cells. And it also leads to evolution, as we're going to see later when we start talking about some population genetics stuff. This mixes up all of the alleles and creates unique offspring from their parents. So humans have 22 pairs of autosomes and 1 set of sex chromosomes. That's an important thing to know. So you have 23 pairs of chromosomes. Most of those are called autosomes. 1 set is called sex chromosomes. And females have 2 X chromosomes, but males have 1 X and 1 Y. Right? So each haploid cell has either an X or a Y in males. So when males are making sperm, they're either going to donate an X to that sperm or a Y to that sperm. And because of that, sex is determined by the sperm, because women or females only have an X to give away. And let me show you kind of what I mean by that. Here's a picture of the chromosomes, by the way. Here's an X chromosome. Here's this puny little Y chromosome down here. There's not a lot of stuff going on. And we're going to talk about the significance of this chromosome later on in the summer or the semester. But X is big, Y's are quite small. So you have this process, this meiotic process happening in both males and females, and this is simplifying it. This is suggesting that you only have an X and a Y, but the reality is you have 22 other chromosomes happening here. But as you can see, the mother only has X's to give. She doesn't have a Y to give. And so in order for a child to become male, the father, the sperm that happens to fertilize that egg, has to be a Y. The sperm has to be a Y in order for the son to get an XY genotype. In order for the son to have a Y gene, he has to get it from his dad, because his mom doesn't have one to give.